Good morning, everybody. My name is Giulia Agostinetto from the University of Milano Bicocca in Italy, and I am very happy to be here presenting our work with the title SKIOME project, Lead Skin Microbiome Research Towards Data-Driven Approaches. The skin is a peculiar ecosystem with a biodiverse microscopic life and diverse ecological niches, where cooperation between the microbiota, host skin cells, and the immune system is responsible for maintenance of skin health. And a disruption to this delicate balance, such as by pathogen invasion or a breach in the skin barrier, may lead to impaired skin function. Thus, the skin can be viewed as a forest, where complex structural interactions sustain the homeostasis of the environment, and perturbation can exert different degrees of effects. This is even more important if we consider that humans are holobionts, microorganisms composed of the host and the interacting microorganisms, which together form a discrete ecological unit. The last decade has brought renewed interest in the complexity of microorganisms living in association with hosts and interacting with the environment. Thanks to sequencing technologies advances, initiatives such as Human Microbiome and Earth Microbiome projects favor the collection of skin microbiome data. This yielded a number of new empirical results, data, and concepts. However, this huge amount of data lacks harmonization and is far from being completely exploited in its full potential to build a foundation that places microbiome research at the nexus of many subdisciplines within and beyond biology. Thus, urges the need for data accessibility and usability according to FAIR principles: findable, accessible, interoperable, and reusable, as supported by National Microbiome Data Collaborative and FAIR Microbiome. The reuse of data obtained from sequencing technologies can enable microbiology researchers to ask new questions and develop new strategies to study the skin and associated microbial communities. Guidelines and tools have been taken to promote standardization of sample processing, sequencing, and data analysis across the microbiome field. However, achieving global standardization is not an easy task, given the rapid emergence of new technologies and specific methods related to different sample types. In order to enhance data-driven strategies, meeting the emergence of making data accessible and reusable, we launched the SKIOME project with the aim to tackle the challenge of accelerating discovery and advances in skin microbiome research. In particular, we collected datasets from the 3 public genetic repositories, which are members of the International Nucleotide Sequence Database Collaboration, the Sequence Read Archive, the European Nucleotide Archive, and the DDJ Sequence Read Archive, which together collect the majority of the publicly available datasets obtained through sequencing together with their associated metadata. We focused our search on the human skin biome and collected, integrated, organized, and harmonized the datasets of interest, trying to achieve a comprehensive human skin microbiome collection enriched with detailed metadata information. To reach this goal, we started carrying out a 3-step data retrieval combining an automatic strategy with a manual curation approach. The first step involved the use of the SRA-DB package in order to collect the datasets related to this field. In parallel, a manual search was performed considering microbiome studies with the following characteristics: datasets belonging to the human skin biome obtained with a 16S sampling on sequencing and submitted from 2012. The second step was dedicated to the enrichment and integration of the metadata associated with the datasets found, combining SRA-DB search and Entrance Direct tool, which provide access to international nucleotide sequence database data to collect the metadata associated, plus a manual research to retrieve metadata that can only be collected from the related publication. Finally, the third step included the removal of redundant and useless metadata and a manual correction of metadata wrongly assigned. This process led to a comprehensive data frame of the studies involved in the human skin microbiome field, enriched with their metadata information. Here we show the results obtained. The manual search recovered 68 studies, about 22,000 samples. These datasets have been manually validated and information on the associated study has been collected from the publications. The automatic search instead returned 203 studies, about 82,000 samples. Even though the automatic search with SRA-DB recovered a greater number of datasets than the manual retrieve, it failed to identify 30 of the manually retrieved datasets. This indicates that the automatic approach cannot yet be used as a substitute for the manual investigation, and neither of the two methods can be regarded as comprehensive. In addition, datasets recovered by the SRADB method lack a manual curation step, but it is necessary to remove undesired studies or samples. After the identification of the studies of interest, we proceeded to collect the metadata associated with the samples on the public databases. SRADB collected up to 70 metadata, while Entrance allowed downloading the information on each given run up to 37 metadata. Overall, we obtained two main results: a list of 81,000 of samples with integrated metadata thanks to the framework used, and a list of 22,000 samples with 50 manually curated metadata. In this presentation, we will show in particular the final curated data frame, including manually recovered datasets together with their metadata. The data frame presents the following structure: about 22,000 samples for 68 studies with 50 metadata associated. 13 metadata are manually collected and contain study-specific information, including sample collection method used in the study, 16 sRNA upper variable region sequenced, the clustering method used in the study, the number of OTU or ESB retrieved according to what reported in the publication, the database and the version used to assign taxonomy, the skin condition investigated in the study, the geographic location of the samples collected, the presence of magnify analysis, and the links of the publications. The other 37 metadata information are obtained from Entrance and SRADB and include information on the study, on the experiment, on the runs, and on the samples. From the manual curated analysis, we had a mean of 323 samples per study. Most of them were obtained from V1, V3, or V4 16S hypervariable regions. The Illumina sequencing platform, in particular MiSeq, was the most used, followed by 454. Considering the type of sample collection method, the preferential protocols were swabs, followed by biopsy method. Considering the bioinformatic analysis, OTU clustering was the most used, combined with a taxonomy assignment with Green Genes database. We decided to investigate the geographic provenance of samples, considering that skin microbiome can be affected by the geographic area, as it is influenced by both the genetic and environmental context. The studies investigated were mostly collected in the United States for a total of 22 studies and 13,000 samples, followed by Europe countries, 19 studies and 3,000 samples, and China, 11 studies and 3,000 samples. Finally, we investigated the skin condition in the studies, mainly thanks to literature review. A total of 26 different conditions were found. In particular, the most abundant conditions were healthy skin with 11,000 samples and 26 studies, atopic dermatitis with 6,000 samples and 8 studies, skin lesions with 1,000 samples and 7 studies, and psoriasis with 600 samples and 5 studies. In general, our work resulted in a state-of-the-art assessment focused on human skin microbiome data and project, with special attention on data collection, data generation, and analysis. Metadata curation was considered a pivotal step to enhance data reuse and exploration. The output of our research will help to speed up future works interested in reusing and integrating human skin microbiome datasets. In addition, this metadata analysis reveals a not homogeneous representation of skin microbiome condition, considering its complexity and the influence of all the actors such as environment or the health of the subjects. This work can bring improvements to future research by having built the basis for knowing what we are currently missing, providing new interesting directions into how metadata associated with microbiome studies should be deposited and how information can be integrated using different sources. Of course, a lack of standardization during metadata completion and data submission still exists, and for this reason, our future work will be focused on improving metadata enrichment and curation, suggesting guidelines for this field of study, aiming to build a refined collection of harmonized datasets ready to be integrated into microbiome projects, with the idea to include them directly in advanced post-processing analysis, such as machine learning or data mining strategies. If anyone wants to know more about the project or wants to get in touch with us to improve our investigation, please feel free to contact us. In particular, here I show my email and the contact of my colleague Antonia Bruno. I would like to thank the International Society for Computational Biology and conference organizers for giving us the opportunity to present the project. I thank our University of Milano Bicocca, the Zooplant Lab Group, and the TechSB PhD program in which I am involved. Of course, I thank all my co-authors for the support, and I thank you all for your kind attention.